Welcome back, everybody. I hope you all had a great lunch. Our next speakers really move away from enzymes and transposons and take us to a higher level of organization, to organisms and populations, and the changes they face to acclimate and adapt to pressures in the environment. And it's my great pleasure to introduce our next speaker, Dr. Joanne Chory, who joins us from the Salk Institute, where she is a professor and the director of the Plant Biology Lab. And Dr. Chory got her bachelor's degree from Oberlin in Ohio, and then went on to do her PhD work at University of Illinois, Urbana-Champaign, and then went on to do postdoctoral work at Harvard Medical School. And what is really tremendous about Joanne is she really has this systematic approach to tackle how do plants respond to changes in their light environment, and what are the genes regulating this activity and their ability to sense and acclimate to the ever-changing seasons. Joanne has done tremendous work in terms of scientific outreach and been a wonderful director and mentor to over 100 students that are now continue to be in academic science. And she's received countless awards, as well as served on many advisory editorial boards. And most notably, she recently received a Breakthrough Prize in the Life Sciences in 2018. And today, Dr. Chory will share with us her current work on how plants adapt to changes in light. And as alluded by her title, 50 Shades of Shade, hopefully the mechanisms enacted aren't anything like the movie.
Yeah, plants have kind of a wild life.
Thank you.
Okay, thank you. Okay. Well, thanks a lot for having me. I'm really having a great day here today. It's been really interesting to hear what everyone says so far. Hopefully you'll like what I have to say too. Okay, let me do that. All right, here I go. So plants can't move, and they haven't evolved a brain, and they don't have an adaptive immune system. Yet there are 350,000 species of flowering plants on our planet right now, and they inhabit every single part of the earth, And this is because they're the masters of adaptation. And so they have a lot of plasticity, and they use it. They use it within a species and between species to adjust to this ever-changing environment they find themselves in. And because they're photosynthetic, plants are really interested in light, and light has a lot of information. And so I'm going to show you a couple of examples. Here we go. Okay, so in this first panel, on the left there, we're looking at Arabidopsis, which is this plant that we study in my lab. It's a little plant in the mustard family. And this is a plant, these are genetically identical seedlings. And they're about 5 days old. And the one on the left hasn't seen any light, and the one on the right has been germinated, and the seed was germinated in the light. And you can see how the body plants already have adapted to those kinds of environments, because the plant on the left that looks like a sprout, that plant is adapted to life under the soil, right? So it's trying to forage its way up and find light so it can become photosynthetic. If it doesn't find it by about day 5 or 6, this plant will senesce and just die. This guy, on the other hand, doesn't have this apical hook protecting those cotyledons. It's open, and it's going to grow, and it's going to complete the life cycle. And so that's a plant responding to the amount of light in its environment. And in the middle, I have another rosette plant called Rudbeckia. And Rudbeckia will flower. It'll stay in the juvenile phase like the plant on the right. until it sees a long day photoperiod. And when it sees a long day photoperiod, one long day is enough to induce it. Oops, sorry. I don't know it's going to do that. Okay, to induce this plant to bolt, and then it goes through the reproductive phase, and then it dies then. And on the right is what we're going to be talking about today. And these are a cartoon depicting a plant grown in bright white light that has the full spectrum of all photosynthetically active light, and it looks like this, say, in this case, some sort of bean, I think. And here's a plant that is doing a process that I call shade avoidance, and it's called that by the field, actually. I'm not the only one who calls it that. But anyways, shade avoidance is an adaptive response, and it's when plants get in competition with each other for light, and these are what the sun-loving plants will have this as a response. And so let me show you how plants, here's a nice example, I thought, of how plants adapt when competition for light is strong. And so on the upper right-hand corner here is a plant growing out with no canopy on it or anything. It's not competing with anything for light. You can see this plant is short, it's branched, this tree is short, branched, and bushy. And over here on the lower left is plants that get too close together. They're all trying to get to the top of the canopy. So these plants are all very similar. They're all the same species. I don't think they're genetically identical. But anyways, you can see these guys are trying to, they've elongated this and they're growing tall. And here's a guy in desperation mode. He's like trying to find light anywhere. He bends over and he grows up and he'll eventually find light too. Okay, so how does the plant know what kind of light it's in? What kind of information is it looking for to know it's in the shade of another plant? It's actually looking at the color of light more than the intensity, though that does come into play when you get under a canopy. And so there are about 13 photoreceptors in Arabidopsis that helps that seedling emerge from the ground and like grow and establish itself. So there's a red, far-red photoreceptors called phytochromes. There's 5 of those in Arabidopsis, and then there's a whole slew of blue light receptors. There's 3 families. Sorry. Here we go. The cryptochromes, which are all throughout the animal kingdom as well. Phototropins, which are unique to plants, and these have a flavin, and they also have a kinase domain. And then the Zeitlooper family, which seems to be more or less dedicated to the circadian clock, but all of these feed into the clock, actually. And there's even a UVA photoreceptor that's not just like involved in DNA damage, but it actually tells the plant when there's that wavelength of light there. Okay, so on the other end of the spectrum, so the photoreceptors are telling the plant, this is what your light environment looks like. All of them are signaling in, and something has to integrate it somehow, and then have a growth response, or not. That's basically all plant behavior, you grow or not, and I think that's pretty much it. And so that's simple. And these are the compounds that plants use to grow or not. And so there are a bunch of small molecule hormones, and 3 of them are known to promote growth. And there are these 3 here. There's the steroid hormones of plants called the brassinosteroids, and the most active one is called brasinolide, and this is something that my lab worked on. We took a 15-year detour down that street. And we know a lot about them now. And then there's the terpenoids, the gibberellins, which are also involved in promoting growth. And then there's auxin, which is a small molecule involved in growth as well. And then those are actually counteracted upon by abscisic acid and ethylene, which is a gaseous compound. So together, these guys tell the plant how big it should be in a certain environment, or if it should have a long stem, or grow leaves, or say restrict roots, or something like that. Okay, so now I'm going to get to the shade. I have to tell you a little more. So shade avoidance is triggered by a change in the ratio of the red to the near far red light in the plant's vicinity. And indeed, a plant actually knows this is a full spectrum white light coming through right here. And here's a plant under, this is what comes through a canopy. So you can see, so the chlorophylls and the carotenoids and things take everything out. Sorry. This was way too far back. How did I get way down there? I'm sorry. All right. And so you can see there's virtually no blue or red light coming through. And so on here, I want to just show you some of the other information the red to far red ratio of light can give a plant. So it tells the plant if it's the middle of the day when the red to far red ratio is high, so 1.19 at sunset, it goes down. That's a little bit, but a plant can anticipate nighttime with that small change in that. Moonlight actually has a lower red to far red ratio. If you're in a deep canopy, you can see you basically have almost no red light because the chlorophyll's up above you, taking it all out. And even in soil, it's getting pretty low. And so shade avoidance actually is an adaptation that has to happen right away. So in Arabidopsis, the plant basically has 24 hours. It adjusts its whole growth habit by doing these things you can see here. So the leaves, first the leaves go up, then you have increased internode extension. This is the internode right here. This is not Arabidopsis, but it's some sort of pea plant or something. I don't remember what it was. And then you have an increased petiole, which is this tissue right here. So everything is just trying to elongate. And then as the day goes on, this all happens within a few hours. It starts to happen anyway. And then as the day goes on, you'll see increased apical dominance or reduced branching. You get reduced leaf area, reduced leaf thickness, reduced chlorophyll synthesis, accelerated flowering, and an increased susceptibility to herbivores. So everything starts going south, and you're not very fit. But what this plant is trying to do is just set some seed and hope its progeny is not in such a shady environment. But the really cool thing about this red to far red ratio that the plant is seeing is that before it's even in the shade, a neighboring plant gets in its face, right? And so far red light starts reflecting off the cell wall of that plant. And it's that change. So that plant is still seeing the normal amount of red, but the amount of far red has now gone up. And that's the trigger that triggers a plant into this competition. It has 24 hours to either beat it or it's stuck in that mode. You can't reverse shade avoiding behavior after 24 hours in Arabidopsis. So that's kind of an interesting point too. Okay, so we wanted to study this. We went back to first principles and we said what would be a good tissue to do a genetic screen in, because we don't really want to do them on those big plants because that's way too far into the life cycle. And so what we decided to do was look at seedlings, and we've done a lot of seedling screens in my lab. And so why did we pick this hypocaudal elongation? The hypocaudal is the embryonic stem. And so here you can see it's a simple structure in either the light or the dark. And it elongates, when it gets longer, it has no cell divisions. It's all elongation growth. And it's a relatively autonomous trait in that tissue. And it's early in the life cycle. So these plants are like 4 or 5 days old. We can mutagenize our seed, plate them on plates, and then we can throw those plates out after we pick mutants out and not doing something, right? And this is very easy to quantify character, and it's exquisitely responsive to the environment. I'll show you at the end of my talk that it's also the way the plant responds to increases in temperature, which we've been using to try to understand their interaction between the light and temperature environments of the plant. Okay, so hypocaudal elongation shows an inverse log linear relationship with the amount of light. So this plant has seen the most light, this plant has seen the least. And it's very quantifiable character. We've done a lot of that kind of genetics, multigenic genetics, because we can make a lot of these measurements. Very nice trait in that sense. Okay. And so say you wanted to, is it a good trait for shade? And the answer is yes. So here's Arabidopsis again, growing as a seedling, about 10 days old here. And you can see in white light where it has the full spectrum of light, the plant's short, these leaves are nice and big, and here I go again. Okay. Okay. So like I short, and we put this guy in the shade by adding far red light to a blue-red light kind of spectrum that they were seeing before, and you can see that plant will elongate. And it does all this within a day or two, so it gets a lot longer. Nice trait to look for. So if you're looking for shade avoidance, what would you expect of your genetics? Again, simple. If you're a mutant and you're positive regulated, that means you can't elongate in the shade. but wild type will elongate on the left and the mutant will stay short. So that's a pretty easy screen, right? You don't have to know a lot of anatomy of the plant or anything. And so, because I don't, but anyway. So here's wild type again. Now this will be a negative regulator. In white light, the mutants will be long. Oh, sorry, you can't hear me. I'm sorry, okay. All right, so that's our genetic screening. We already had a mutant that qualified for the negative regulator. And so let me tell you, so that mutant has something to do with photoreception. And it's one of the phytochromes, not all of them, just phytochrome B. And so here's the reason why you would get that out of a shade screen. Because here's the spectrum of chlorophyll on the left. And you can see chlorophyll has an absorption maximum at 660 nanometers. So phytochromes have evolved with chlorophyll, okay? Because they have a long-lived spectral form called PR that absorbs at exactly 660 nanometers. And then that gets photoconverted to another long-lived form in their photocycle, the way photoreceptors do. And that is called PFR. That's the active form of phytochrome. And that will go on and promote a lot of processes in plants. So those pigments evolve together, the pigment that's part of the phytochrome, the whole protein, with chlorophyll. And that's how a plant knows it's in the shade of another plant. This is the photoreceptor that's going to tell the plant that. And here's how we know, because when we had phytochrome B mutants, it's them growing in white light with wild type over here, and here's white light, here's the unshaded and shaded wild type. And you can see 5B looks a lot like a plant growing in the shade when it's not in the shade. So I think that's it. So that was our first mutant. It was the negative regulated pathway. Its job is to tell the plant, you're not in the shade, so just stay short and bushy. Okay, so this plant always thinks it's in the shade when you basically take out phytochrome B. Okay, so then Etal was in my lab. This was a long time ago. I'll go through this quickly. But she got the first shade avoidance mutants. And this shows how she did the screen. And so here's our white light conditions. We're growing the plants in red plus blue. That's photosynthetically active wavelengths of light. And then we add the far red light in an LED growth chamber. That's simulated shade for us. It's a red to far red ratio of 0.6 if you really care. And so what that did was separate the plants into two populations, the seedlings. So you can see here, okay, here's the short one in the white light, and here's the one here's the average of the plants that come out in the shade. So it's pretty separate. So we thought we had a pretty good screen. We want to get a lot of false positives. We got 3 loci out of the original, out of the original screen. I don't have a, sorry, I'm moving a lot. Anyway, and we had 3 alleles at this one locus called SAVE3. Here's this phytochrome B mutant. These are all minus and plus shade. And so you can see here, wild type gets longer. These 3 alleles get a little longer, but they're not really all that long. And here's this 5B. It's just long all the time. Okay, so E cloned SAVE3, and this is what came back. So you've cloned an allanase. And we go, what the heck's an allanase? We didn't know what that was. And so allanases are the enzymes that are responsible for giving garlic and onion their smell. Okay, so it's not exactly what we thought we'd get out of a shade avoidance screen. And so we went and talked to Joe Noel, who's a colleague of ours at Salk. The guy's really good biochemist, knows a lot about secondary product pathways. And he said, well, it's allanase maybe, but it is just an aminotransferase. So now you have to just figure out what's the amino acid that it wants to change. And so E did that with Flo Posier in Joe's lab. Oh, wait, while she was doing some of that stuff, we were also looking at what this plant's defects were, okay? So this is just the growth curve of wild type of Arabidopsis, which is called Columbia. And here's some mutants that people thought might be in a shade avoidance pathway. They're not really in it. And here's our mutant, SAVE3. And you can see, so there's 2 growth phases. There's a little blip right here. And there's a lag before it, and there's a little lag after, and then there's a big growth curve. This is all within the first 8 hours that the plant is in the shade. So you can see there's quite a lot of growth. SAVE3 doesn't do it. What else could we find out about SAVE3? Well, it turns out SAVE3 actually accumulates pretty high levels of auxin when it goes into, it doesn't accumulate high levels of auxin when the plants are put in the shade. And that was actually a surprise. So the wild type, no one had ever made the measurement. So you put wild type in the shade, and it makes twice the amount of auxin that the seedling used to have. That's a lot when you're just taking the whole seedling like that, because we don't know where in the seedling it's being made. And so SAVE3 didn't do that. So that's the first hint that it might be in the synthesis of this plant compound that's known to be a very important hormone in plants, called IAA or auxin. Okay, so she wanted to look at that a little more. So the stem is what grows, but is the stem where the auxin might be being made? And the answer is shown here, not the best slides, but the bottom line was that this DR5 Gus, which is this reporter in this background here, is an auxin reporter. It goes up in the cotyledons, which are the leaves, right? Not the tissue that's growing, which was a little bit confusing. Then we used an inhibitor of transport of auxin, which is known as MPA. And you can see if we keep the IAA in the cotyledons of the leaves, the plant doesn't elongate. So that IAA has to get out of there and down to the stem. That's an important part of what's going on in shade avoidance, it turns out. And so back to the biochemistry with flow. And so now they showed, we had a hint was auxin. So where would auxin come from? It turns out the auxin biosynthetic pathways were not known, even though this is like the most important plant hormone. But anyways, it comes from tryptophan. Jerry Fink's lab worked on this for a while, but they never really figured it out. I think they kind of gave up, and people moved on to other stuff. And so here, it's trypaminotransferase is what SAVE3 is. So that was cool. So now we say, okay, so maybe it's the first step from this pathway of TRP. It's like dopamine coming from tyrosine in a way, right? And so, okay, so now we have that. So maybe we can hook it all up to this change in the light environment, and then we see a change in auxin. Well, we couldn't show regulation of the RNA. The protein didn't accumulate when we put the plants in the shade. And we tried a lot of it. We tried overexpressing it. It's not really the rate-limiting step of whatever that pathway it might be in. But anyway, so we just kind of kept all that in mind. We didn't really know how it was regulated by light. We couldn't hook the changes in morphology of a shade seedling to a difference in the photons that the plant was seeing. And then we finally figured it out. There were some old mutants that we had, and this is the reason why no one ever figured out how auxin was synthesized in Arabidopsis. Because SAVE3 is one of 5 trypaminotransferases. They all can make auxin. This takes out about half of the total auxin the plant makes. So SAVE3 mutants in white light had no phenotype. So if you have half the amount of normal auxin and the situation is good, you don't see it. What the shade did was give us a sensitized mutant screen. And so we were able to see the effect of losing just one of those. So that's SAVE3 is the first step. And then IPA was a known intermediate, but people didn't know how you got there necessarily. And then anyways, this second step turns out to be this category of enzymes called YUX. And the YUX has 11 of them. They're all redundant. They all make auxin from IPA. And so we had found that in a gain-of-function screen about 10 years before we found SAVE3. And so we think this is involved in auxin because we could make long hypocotyls when we overexpressed any one of those in the stem. But we didn't really know how it fit. But it turns out this is the whole pathway. And we were able to show this with Yan Zhao, who used to be in my lab, and Joe Noel, who's a friend of mine, and a good collaborator at the Salk. So this is the textbook pathway now. Yay, so we filled in a big knowledge gap, but we still haven't linked a change in photons to, a change in the architecture of the plant. So back to mutant screens. Okay, so how does auxin growth connect to the photoreceptor? All right, so there's a family of BHLHs that was known to interact with phytochrome, so we just started to look at those as mutants. And it turned out there was one called PIF7, and PIF7 hadn't been worked on by anybody, and when you put it through the test, it's a shade avoidance mutant. See, here it is, white light on the top. There's Columbia, the wild type, PIF7, 2 alleles. And you can see this is work of Lynn Lee that came later from the first stuff. And so PIF7 looks like it's a shade avoidance mutant. So does it have similar traits to, say, SAVE3? And the answer was yes. So here they are tracking growth together. On the bottom here is SAVE3 and PIF7. And you can see they're both missing that first blip, and they have a very reduced second growth phase, so the plants are both short in the shade. Okay. They don't make much more auxin when they go in the shade either. They make a little more than SAVE3 was making. But in general, wild type makes a lot more, and you see that phenotype, but you don't see the elongation in PIF7. And then we looked at, we did, I think these were microarrays at this time. But anyways, here's the PIF7 versus SAVE3 versus wild type. And you can see these two look alike, right? So this is PIF7 and SAVE3, or vice versa, PIF7, SAVE3, in white light and then in the shade. They're very similar. And so about half of those genes that changed their expression were known auxin-regulated genes from just treatments of auxin on the plant. And so we looked at those and then we said, well, who might not be regulated right? Well, it turns out 4 of those yucks are actually misregulated in those mutant backgrounds, both of them. And it's not until you knock all 4 of them out that you see the shade avoidance phenotype. So you have to be patient in Arabidopsis because everything's so redundantly encoded. Anyway, but I think that is sort of the end of the story. And I'll just summarize the rest of it. The expression of these 4 genes is upregulated in shade, in wild type, but not PIF7 or SAVE3. G boxes and the promoters of these are responsible for their shade-induced expression by just PIF7 that binds there. We knew what the element was. The chromatin IP confirmed PIF7 was bound in vivo. And finally, this quadruple mutant was made by Julian Malouf at UC Davis, who used to be in our lab a long time ago, and also works on shade. But he was onto this too because he had done similar experiments with microarrays or RNA-seq. And so he was nice enough to tell us that result before he published it. He's published now. Okay, so we have a pathway. We filled in the whole thing. It took us almost 20 years to finally fill one of these light pathways in. And we had to fill a big knowledge gap, which is why I think with the whole auxin part of the story, which was unexpected that it would be something like that. And so here's the model. So in the open canopy, you have 5B, and 5B gets irradiated by red light because it's not being blocked by chlorophyll from another plant. That turns into PFR. PFR then moves to the nucleus from the cytoplasm, where it interacts with PIF7 or other PIFs, and then they get degraded, and that allows a growth inhibition response. So when PIFs are bound, you get growth promotion. Okay, so in the shade, you're going to get growth promotion. And so 5B doesn't go into the nucleus because it's not really there or functional. Or it's not turned to PFR because the red light levels are so low. And so you get reduced, at least a reduced amount goes into the nucleus, where they would interact with, it's not going to interact with PIFs now. And then you get the growth genes turned on. So that is how you get growth in the far red part of the spectrum. But that doesn't tell us what's happening in the stem. Okay, so that's all what's happening up in the shoot, up in the green part of the plant. So then auxin moves down. You can see this is this auxin reporter gene, the DR5 promoter. So this shows you 5B is being expressed everywhere. But auxin is only being turned on in certain cells in the cotyledon. So you can see it's only those cells that are on the leaf edge. So, I mean, it's not even though cytochrome's in every cell, it's not going to make auxin everywhere. And then that auxin moves down into the stem. You can follow it. Okay, so our model for shade avoidance has some predictions that the auxin has to move down, okay? So here's one. Removing the cotyledons will inhibit hypercotyl growth because if that's where the plant has to actually increase auxin and then send it down, it has to happen in the cotyledons. So we took them off and just irradiated the bottom part of the plant. And it didn't work. So removing the cotyledons did inhibit. It didn't. They didn't grow. So we gave that a star. And then we said, what about if we should have a gradient of auxin responses away from the cotyledons in this stem because we have all these readouts of auxin-inducible genes. And that got a star too. Okay, and then another experiment. These are all done by Carl Procco, who's still a postdoc on my lab. He irradiated the cotyledons with low red infrared light, and that should induce the hypocotyl to grow. So he blocked light from the bottom, and when he did that, that was enough to make the plant grow, and when he irradiated only the stem, it didn't grow, not the cotyledons. So all these say our model's probably good. And so how is growth of different cell types coordinated by auxin in that stem, right? So here's a cross-section through the stem. This is work of Carl again. So he cuts the stem. These would be the cell types you see down there. And so the auxin response pathway is known, and its receptor is known, and it involves a repressor. You can make a mutation that blocks auxin response in these small proteins called the AXRs. And so what Carl did was he had a promoter for each cell type down there and expressed that dominant block in all these cell types. And here's the experiment. And so here's an endodermis-specific one, for instance. Here's the green tissue in the middle. And here's an epidermal-specific promoter. Blocked the response in all those tissues, and it only mattered in one, the epidermis. So somehow that auxin comes down, and it has to get out to those cells. What people think plant growth is and elongation growth is that the inner tissues push, and the epidermis is restricting, okay? So that's somehow, if they keep pushing, and the epidermis allows it to happen, so that's how you grow. It's generally how people think about it. And so you have to have an auxin response out there that's going to loosen those cell walls and allow those inner tissues to push out. And that's going to allow you to grow. Okay, so Carl asked, is there something specific about epidermis that, that's different in how it responds to auxin? And he found that it was, and this is like the epidermis-specific RNA Snake experiment that he did, where he peeled off the epidermis. You can peel it off with little number 5 tweezers, And so anyways, and then he protoplasted those cells. And this purple part here are these genes called SOURs. And SOURs are encoded by a very redundant gene family. And they promote growth, auxin-induced growth. And they're all on, and these are all ones that promote growth and are on in the epidermis. And so they're probably how auxin is promoting growth in the epidermis. But that's still not the whole story. Because when we hit that big growth curve, something else was happening. We did a time course and we took tissues apart. And what we found was that right when the plant's about ready to have the big growth response, it turns on the whole brassinosteroid biosynthetic pathway. And so these are the brassinosteroid biosynthetic genes. Most of them turn on, and they mostly turn on in the hypocotyl. And so this is important, actually, because we knew brassinosteroids don't move much in the plant. So the cells that are making them are the ones that respond to it as well. And so what this is telling us is that the brassinosteroids need to kick in and help auxin out at that point. And we already knew brassinosteroids act to promote growth in the epidermis. So they're both acting in the epidermis at the same time, and then you get a lot of growth. Okay. And this just shows if you have a mutant in one pathway, like the BRY1's receptor for brassinosteroids, you can't make auxin promote growth. So they both have to be functional. That's the genetic experiment that shows that. And so these two hormones act together to promote growth in the shade. So that's the story mostly about red light-induced growth. I'm going to skip through this, but it turns out Gibberellins, which is another hormone involved in elongation growth, plays a role as well. But we don't know if it's happening in the epidermis at this point with that tissue. So anyways, this just shows that you turn over a protein when you grow, and Gibberellins are the receptor that's responding to that growth signal. And so you get growth promotion when that protein goes away on the left. And so now we have 3 plant hormones involved in promoting growth of just 20 cells, okay? And so you have this small little growth response. It involved multiple tissues and many different hormones that were acting non-redundantly on the process. A lot more difficult than we were expecting. All right, okay, so here's our pathway in the shade so far. This is just one kind of shade, all right? So we make new auxin up in the top part of the plant, we transport it. We don't know if it's regulating GA biosynthesis or signaling, but somehow that gets involved. That involves, okay, sorry, I'm going to sit now. All right. Okay, and okay. And so auxin is required for that delta protein to turn over, and you have to make new BR and BRX locally in the epidermis, and then you shade avoid. So that's the story there. And so, and I want to just tell you about, very quickly, not in that kind of detail, two other ways that we made shade. One of them was by diminishing the blue light part of the spectrum, and one was just taking out the total photosynthetic light, which is what a canopy does when it closes. Okay, so our question here was, is the growth pathway going to be the same as the one that everybody always studies, the lowering of the red to far red ratio of light? And the answer is no. And so auxin doesn't even appear to play much of a role in diminished blue light. And so the photoreceptor is different too. So this is kind of some data that says the photoreceptors are, this is saying the PIFs are involved, these BHLHs, but it's not really PIF7, it's these two other PIFs, PIF4 and 5, and it involves cryptochrome. So they interact with cryptochrome. Oh, this is, again, more PIF. So PIFs accumulate when you take the blue light out. So we're selectively removing blue light from a broad spectrum of light. And so especially PIF5 accumulates at pretty high levels. It's accumulating in the right places. Sorry. All right. Okay. Sorry. I think we better put this up on this here. No, all right. And let me get through this. And this is the photoreceptors. The cryptochrome 1 and 2 both are involved. And so this is a response now to that kind of shade that's telling the plant you can't really do photosynthesis very well when you don't have blue light. And so it involves cryptochromes as the photoreceptor, and it involves two different PIFs, PIF4 and PIF5. Okay, and so we looked at the Rna Snake Experiment. We did the same kind of time courses. And what we found was virtually like almost no overlap at all. So we had low red to far red light. It involved all these auxin-inducible genes. Half the genes that were involved were auxin-regulated genes. We had very few of those in the blue light. Instead, what we saw was a whole bunch of cell wall loosening enzymes. So it looks like blue light might be acting directly on cells, and just loosening up the cell wall and allowing the plant to elongate. And so now we have these two pathways. And then this shows that PIF4 and 5 are bringing the cryptochromes to that on a ChIP-seq experiment, sorry. And so we knew the binding site for cryptochromes and for PIFs, and we could show that with the right mutants and all this other stuff with ChIP-seq, that the PIFs are there, and then they bind cryptochromes, and they give you a certain response to that kind of wavelength of light. Now, those two PIFs also bind to phytochrome, not in the same place, though. They use a different part of PIF to interact with cryptochromes. So that was kind of a surprise, too. So both the red and blue light pathways are using these PIFs. The PIFs are the ones, if they're either there or not, you grow or you don't grow. They're really important to the environmental input to these developmental pathways involved in growth. Okay, so then last experiment, we lowered the whole, the whole red and blue part of the spectrum. So the plants are basically starving. Oops, sorry. Okay, so here you can see we took out the blue part and the red far red. And this was mostly done in Christian Fankhauser's lab in Lausanne. Okay, and so what he was able to show, so here's the three pathways now. So in low part, we don't know the photoreceptor, but auxin levels stay low, and they're really even lower than in wild type. But the sensitivity to auxin was high in these plants, so they somehow can elongate their hypocotyls. And we don't really know how it's modifying that signaling pathway directly, but it seems to be somehow. And so we have the PIF7 pathway for the red far red, and PIF4-5 involved in the blue light part of the spectrum. Three pathways, more or less, but some similarities. Okay, so this whole thing about the top of the plant being involved in perceiving light, when we knew the stem can make auxin itself, but what the plant is doing, it makes these conjugating enzymes in the stem, and it keeps the level of auxin low in the stem, so so that the stem is a sink for new oxygen coming in, so the plant can have the flexibility to grow right away, I think. And so this is what I decided is going on. So if you fire red light and you're coming in from the top, that's just a reflection, that you're shading yourself, all right? So plants, cotyledons are always, the leaves are always shading the stem, but that's not the same as being in a competition with another plant. And so we think that when the far red light starts coming in from the side, or some angle from that side, the plant needs to distinguish it. So it involves the leaves, and then somehow, that's when you do the competition far red light growth. So that's what we're thinking right now, and you can actually irradiate from the side, you can get a nice shade avoidance response. So I think we've kind of been doing that more. Okay, and I just want to tell you one last little story. And this is like the high temperature. This is high ambient temperature for Arabidopsis. It's 28 to 30 degrees. And normally we grow it in the low 20s. And this is how it looks. The middle plant is what it looks like when we normally grow it in our lab. But when you put it at 28, you can see it starts looking like a phytochrome type mutant, right? It's like a mutant in the shade, or a wild type in the shade. It starts elongating those petioles. The hypocotyl is long. And at 16, it's even shorter. Okay, so this is work done in Phil Wigger's lab, and where is this? Okay, and in Kara Franklin's lab, both in Europe. It's going to be a really interesting story. So you increase the temperature of the plant, on the plant, and guess what the thermoreceptor is? It's probably phytochrome B. And that's really cool, I think, because it's the rate of dark reversion of Phi B. And when the morning comes, the plant knows how much it has left, and that changes with temperature. And so that tells the plant like what time of year it is. And it's really that integration of temperature with light that tells the plant it's spring versus fall. Because photoperiod is not enough information, right, unless you have memory and know that winter just preceded it or whatever. And so now we have these 4 pathways. So in that pathway, we have the same photoreceptor as shade. Oops, sorry. We have PIF4 as the major PIF involved in perceiving that temperature response. It involves an increase in oxygen, very similar to the red far red changes, and then you get hypocotyl elongation. Okay, so here's a summary. So oxygen's made, here's my summary what I said, oxygen's made from TRIP by a simple 2-step pathway involving a TRIP aminotransferase and an IPA decarboxylase. So we've closed the knowledge gap after 100 years of people trying to figure that one out. So that was really cool. I totally unexpected that it would be like a shade screen that would reveal that pathway to us, but that's what it was. Oxygen plays a central role in rapid growth response to nearby plants. Not all shade is the same. It's the light quality detection versus photon counting shade might be different from each other. So we're going to have to try to figure that out a little better. And high temperature, and also acclimation to cold involves these PIFs as well. And multiple light signals are integrated through PIF 4, 5, and 7, these BHLH transcription factors. And they optimize the body plan and control flowering time in response to the particular environment the plant is in. And growth ended up being a lot more complex than we anticipated. If I have one more minute, I want to show you we're trying to do multiple signals on plants now and see if there's a hierarchy in the response. Can I show you guys that? All right. All right. So here's our ongoing experiment. So we're trying to figure out how you get specificity to each of those PIFs. And then here's this response to multiple signals. And then we want to actually be able to measure these hormones quantitatively with a genetic sensor in the plant. We've been failing tremendously at that one. And then we want to know if we can actually have an impact on agriculture, having this knowledge about how plants shade avoid and they lose yield, which is very important for pig farmers, right? Okay, so this describes some chip-seq experiments that I don't need to talk about. Here's our protocol for doing this multiple treatment. So the first thing we're looking at is shade plus high temperature, which is kind of like a weird situation when you think about it, because shade should be cooler than sun. I don't know. But anyways, that's the way we're doing the experiment. So we go 3 days at 30, 3 days at 21, then we either push them to 30 or keep it at 21, or we put it in 21 plus shade or 30 plus shade. And what that told us is what we already knew first. First of all, so PIF4 seems to be the predominant regulator that acts in the temperature response, whereas PIF7 is the predominant regulator in the shade response. And then when you look at, and so this PIF-Q means it's a quadruple mutant. I couldn't change this on this because it came in as a scan. Sorry about that. But anyways, PIF-Q is the PIF1345 mutant. It still has PIF7, and you can see it can respond to shade. But when you take out PIF7 and the PIF-P mutant, then you can't respond to shade anymore. And so PIF7 really is playing a dominant role. And it turns out if you can't respond to shade, then you can't respond to shade plus high temperature. So that's kind of interesting. It's not just like one and one. So there is a hierarchy. 7 has to be around for 4 to do its thing. And I think that's kind of shown on this slide. No, sorry, I went way over my time. And so that's the end of my story. Here's these 4 pathways. We know they have to interact somehow. They involve similar components, but they give specificity to a particular change in the environment. So it's turning out to be a pretty cool story. It's taken us a while to get there. And then this is to remind me to tell you, the shade avoidance is an adaptive response that's risen many times during evolution in the sun-loving plants. And all these, that's the Arabidopsis on the left, but the rest of these are crops. And that's where I think we can have an impact, is that because you can think about ways to modify the architecture of these plants that will help to increase yield, and we need to do that because we're going to be losing a lot of yield because of temperature increases. And corn, Iowa's not going to be the best place to grow corn pretty soon, unfortunately for Iowa. Okay, so here's like the economic impact of shade avoidance. Here's those 2 soybean plants. The one on the right was planted at high density. You can see how it really has an impact on the yield of those seeds versus the plant on the left. And so you actually get more yield if you plant less densely. But see, corn has already been selected by corn breeders to have a maximum architecture for non-shading itself, non-self-shading. But most of the other crops, no one's even touched the architecture of the plant. So I think we could have an impact there. So anyways, in the last 10 years, these are the people who worked on this project. This is just one of the projects in my lab. And a lot of them are gone now, but it's still the core group here. And we've been funded by an innovation grant from the Salk, NSF, and NIH, and of course HHMI is a big bulk of it. And we've had great collaborations. And at Salk, these are some of my collaborators. Joe Noel, I talked about how we collaborated on the oxen story. Joe Wecker does all the chip stuff with us because we're not really so great at it, but we're getting better. And Saket Navlaka is a machine learning guy who's trying to teach plants, how we're modeling growth. So hopefully that will help us make this a little more simple. And then Steve Kay has been a collaborator of ours. He's been at various places in Southern California. Yander Zhao, a former postdoc in my lab, who did a lot of the oxen story with us. And finally, we have Carlos Baleri. He's an ecologist. I didn't talk about that at all today, but we have some cool results from him. And Karen Lang does all the oxen measurements for us. And I think that's it. Thank you very much.
Thank you, Joanne. Questions?
I have a question here. You didn't say this, but I'm assuming because you have this oxen gradient that the stem is growing in kind of a directed manner, where the levels of oxen are higher, you have more growth. Is that correct?
Yes, that's right. Can you raise your hand one more time?
Hi.
Where are you? Oh, okay, yeah, that's right. That's right. Yep, and so it's not the same for every plant, it turns out. The gradient happens in different directions depending on how big the plant is. Yeah, but yeah, we can correlate the growing part with the level of oxygen. We've done it in Brassica, which is a lot bigger than Arabidopsis because those Arabidopsis seedlings are tiny, as well as Arabidopsis. So.